Howdy, y'all. Welcome to the EEARSS podcast, produced by Terrier TV and sponsored by Edna Wilson with Celebrate Remax Aerospace. If you're interested in buying or selling a home, you can find Edna Wilson over at ednawilson.com. What up, Bill? How's it going?
Hey, nothing. It's the end of a week, and just the end of the week.
Dude, I fried today so hard. It was so hot today. It was one of those days where it was so hot that the sweat's getting in your eyes and you can't see anything anymore because you just got you're just, I mean, you can see my eyes now. I'm all bloodshot.
It's one of those things where there's a front like right above us, and so it's like a wet sponge. It's just awful.
Wet sponge.
Wet sponge. Yep.
Now I can't even think about what a sponge would be like if it was dry.
Well, see, it would be harder.
They're meant to be wet because, I mean, it's a sponge.
That's right.
Technically speaking,
That's
it
the
is a
way they
sponge.
live, yeah.
Otherwise, it would be just, I don't know what it would be, a big fluffy thing.
No, it'd be a rock.
If they
dried out, it'd be a rock.
Dried out's a rock?
Yeah.
Okay. I'm taking your word for it.
That's what I'm going with.
It's a fossil? Maybe it's a fossil.
Maybe it's a fossil. Maybe.
Huh. Okay. Are you ready to jump into chatting with our guest today?
Let's do it.
I'm pretty excited about it.
Yeah.
I'm pretty excited about it.
You've told me. I've never met the man. So
yeah, I've told you stories about him. Yes, this is true. I have some stories. So today on the podcast, we are joined by Dr. Brandon Moore. What up, sir? How's it going?
Hey, Rob. Hey, Bill. Good to talk to you guys.
So, me and you first met in Argentina, I guess in, 2018, I'm guessing?
We did. It's a long time ago, but it was a good time.
Is that when you had the bullet on you?
That is, yes. I don't know if Brandon was there for that whole situation or not, but yes, that is.
I do remember the story.
That is when I, apparently traveled from Florida all the way to Argentina and nearly back with a bullet in my pocket, yes. Yeah, it is what it is, anyhow. So, Brandon, you have a pretty unique journey. You've, man, what have you not done? That's probably where we should start with this conversation. Tell us about how you. Oh, sorry.
I don't do much with mammals. Let's put it that way. I think I like things that are a little scalier, a little toothier, a little more in the wet. So I've never done anything with mammals, but I have a pretty good range of research for crocodilians and turtles and amphibians. I think I gravitate towards that end of the spectrum.
Well, your college career, where did you start off at?
My college career, I started off as actually scuba diving.
Okay.
For research. I was a non-traditional student, went back to school, and I wanted to be a scientist. There was a point when I was a kid that it was like, I want to be a scientist. And I had a little detour on that. And I said, when I eventually said, I got to go back to school, I said, what I'm going to do? And I said, I want to scuba dive. I want to do research underwater. So the first class that I took going back to school for my research career was scientific diving.
Interesting.
And,
hold on a second. Scientific diving.
Yes.
What is that all about?
It is the precision of doing science underwater, of understanding how to measure off transects, how to lay lines, how to use a clipboard, how to interact with your dive partner to gather data. All of the technical skills that you would need to do underwater research.
I had no idea.
Okay. Well, I would imagine like underwater, there's any kind of current at all. All of that kind of stuff is difficult.
It sounds like underwater land surveying is what it sounds like.
I want to tell you, if you want to have a slight idea, it's like being an astronaut. Because once you're down there, you only have what you brought. And you're constrained by the environment. And it's just more difficult. You can't just bop back into the ship and get something, or it's just a very different environment. So learning to work underwater, and probably all underwater welders and other underwater workers know this very much, it's just a different environment to work with. And if you want to do science underwater, this is part of the training to be not only effective, but also safe while you're doing all of this.
That would make you an amazing planner, I would guess.
Yes.
Yeah.
Very much so. And when you're getting it is when you're knowing things, but you're not having to think about them. You know your dive table, your depth, all this stuff, and you have this sense of calm because you're in control in the back of your mind. And there was a cool time in my life where I did a lot of stuff like that.
Okay, well, where'd you jump into after that? What'd you jump into next?
Well, the research was in California, in Catalina, and I was studying this fish called the blue-banded goby. Not a small little pretty fish, but the crazy thing about this is this fish can change sex, and actually very rapidly. They live in groups of one male and multiple females, and the females lay eggs, and the male fertilizes those eggs. Now, it sounds like a great little life. You're living on a rock around an urchin, but what happens if you lose your male? You only have a bunch of females. That's not really functional for a reproductive community. Well, in this fish and many others like it, it's not uncommon. The largest female will rapidly change sex to become a male and replace the male and breed with the females.
Now, how do they? So they just know who the largest female is.
There's a lot of aggression.
I was going to say there's got to be some kind of.
Yeah. There's a social hierarchy, and when the male is gone, the females all kind of do their own little fight club, and the one that wins the fight club.
Okay, the one that exerts dominance. Yeah.
Yes.
Okay.
Yeah. So they'll do what's called displacements. I move towards you, you move away from me. The one that can displace the other ones is going to start this neuroendocrine change.
Wow.
Where their brain changes, their hormones change, their gonads change, their genitalia change. And just think about if it was the case where in 1 week you would change sex. You know, if you were a lady, 1 week later you're a functional male. I mean, that's just if that doesn't make you find biology fascinating, we can't get you there.
No doubt. That's crazy. Crazy awesome. Yeah, crazy
now,
awesome.
Don't amphibians do something similar?
They can have they do not. There's no directional sex change. They do have some organs like a Bitter's organ, which
because I watched Jurassic Park, and I know that.
Okay, now what you're talking about is parthenogenesis. Okay, this is
I don't know what I'm talking about.
Change.
It's ironic that Brandon and I just literally 5 minutes before I got here, the term Jurassic Park came up in another conversation, so this is kind of funny. But
okay, well there you go.
So there's sex change where you change from one binary sex to another. Okay, but parthenogenesis is really not understood, but it's actually more common. And now that we're looking for it, we're finding it. Is where a female. It doesn't happen with males. Males can't do this because males can't make an egg. They can't do all this. But females, left to themselves in many species, sharks, crocodilians, snakes, left by themselves, we're talking about a snake that has never seen a male before, will suddenly produce clonal offspring. I'm not an expert in the genetics, but they will create offspring that are genetically the same as themselves. And you have to think about it because there's no other partner contributing genetic material.
Right.
But imagine you're running a zoo.
Wow.
And there's a tank in there that's a shark. One shark has been in that tank for 10 years. And then you come to work one day and there's another shark in there. and it had young. That's parthenogenesis. And we're starting to realize that is an alternative reproductive strategy in nature. When left to their own devices, some female species can do this. How? What initiates it? That's still to be discovered. But people didn't believe the process. But now the more zoos and other facilities are reporting it, and they're able to do the genetics on the offspring and say, yeah, that is a parthenogenic birth because there's no genetic diversity between the offspring and the mom. Yeah, it's a thing.
Remember originally people were seeing that, they thought people were playing jokes on them and stuff, and it's happened so much now at this point. I've had people try to explain to me that how exactly it happens, and I remember me and Bill talking about this once before. Yeah. I always get so far into it, and man, I think I have a pretty good reach on things and a pretty good handle on it, and then I get about halfway in that conversation, and I'm just like, man, I'm lost. I'm lost.
Yeah, because what would trigger it? What would
need probably?
When does the need? Because you don't know anything. You don't know any better. It's got to be an instinctual thing.
And if is it 8 years, 10 years, 12 years? What's the difference? What is the trigger?
Yeah, and you didn't learn it.
Mm-hmm. Yep.
It's something that's in your hardwired into you.
Yep.
That's wild.
Yep.
Some bizarre
stuff.
But
I think it comes down to one major statement is the women don't need the men. Give them enough.
There you go.
They're just going to take care of themselves. They're just going to say, "Yeah, you're redundant. We're not." Yeah,
we didn't really need a scientist to tell us that. Thank you.
Now there is no parthenogenesis in mammals, but there is in other vertebrate animals.
Crazy stuff.
So
what was next after that, buddy?
Yeah, I was doing the sex change research, and that was undergrad, and I thought, okay, I got to do a PhD now. And this idea of how organisms interact with their environment was really, especially with reproduction, was really fascinating, because here was a fish that changed sex by its social interactions. The next, I was talking to people about where to go, and they kept mentioning this guy named Lou Gillette. I heard it from like direction, from left, from right, from up to down. I'm like, okay, I got to check out this. And Lou Gillette was a professor at the University of Florida, the school in the zoology, and he ran a laboratory that looked at the impacts of pollution in Florida lakes on the mostly reproductive biology of the American alligator. And I thought that was super, super cool. Now, I'd never worked with alligators before. Heck, I've never really been on an airboat, never been in a swamp, so all of that was super, super new. I was on a Zodiac in California off the Channel Islands, doing that sort of fun stuff. And I thought, okay, here's something really new and different. So I started at University of Florida on my PhD, and I was working on ovary development in female crocodilians. So a lot of the development of the ovary takes place after hatching. So the production of what's called a follicle. An egg with a layer of nurse cells around it. In humans and in most mammals, this happens in utero in the second trimester. But in alligators, this happens after hatching, in the first few weeks after hatching, and nobody's really characterized that progression, that morphological maturation. So that's what I started doing for the majority of my PhD there. Doing some testis work, but mostly trying to figure out how an alligator's ovary develops and organizes itself after hatching.
Okay. So I know for a long time you've worked on that and crocodilian reproduction.
Yeah.
Can you tell us a little bit about that research?
Okay, now you're wanting to get to where you want to get to. I was getting towards the end of that, and I was moving on to a postdoc, and the story was I was trying to do a 3D reconstruction of an ovary, where we would take histological sections, slide sections, and then use a computer program to reconstruct the three-dimensional shape of the ovary, of the follicles, and all the things in there. And I had a undergraduate, Keaton Mathavan, and Keaton was a great student. I said, we're going to do this, Keaton. I said, hey, let's start with something, ovaries are complex. Let's start with something simple. And we're like, okay, what's simple? And I'm like, okay, what's a simple cylinder here that we can probably do this 3D reconstruction? Well, I'm like, well, what about an alligator penis? More technically a phallus, because they don't have a urethra. They don't pee through it. They only emit gametes. So more technically to call it a phallus. And I said, what about this alligator phallus? It's a cylinder. It's pretty, we could do this thing. And he's like, okay, we'll practice with this. So we embed this tissue in wax, we section it, we make slides, and we go to start capturing these slides for the reconstruction. And we're looking at the tissue and we're like, wow, this thing is complex. This has some really crazy structures in here. We kind of had this assumption that it would be not that interesting, but the more we looked, the more we realized about the complexity of this structure. And the truth about the matter is, at that time, there's nobody who studied the anatomy and the morphology of crocodilian male reproductive organs. And this is a fundamental of science, is where do you go? You go where no one's gone before. It's kind of the Star Trek of science. Go where no one has. So that brought me into a direction where I started studying the reproductive anatomy of male crocodilians. And again, it's great territory because nobody had published anything of any explicit detail at the time.
And it's super important due to the fact that so many of the crocodilian species are critically endangered too, right? Because you're taking that to the next level as well.
Yeah. There's a basic biology to it, just to understand, to know. And it also tells us about how animals diverge structurally and functionally. So there's some basic biology to it, but there's also another applied side to it. Is so many crocodilians are losing habitat, and their genetic diversity is starting to be taken up by zoos, which have representative animals from these crocodilian species that are losing their habitat dramatically. Well, the problem with that is a reproductive alligator or a reproductive crocodilian, as you know, Frank, is pretty darn large.
Yeah,
And
yes, sir.
not exactly something you just strap into a seat next to you on a commercial flight. So
Yeah.
if you have a female of genetic interest in England or Europe, and you have a male of equally genetic interest that you want to breed in South America, well, it's pretty hard to move from one to the other.
I hear Frontier has flights that are just for that.
You get a companion ticket.
Yeah, exactly. Get a companion ticket, you're gone. You're there.
Oh, Lord.
Did we just gain or lose a advertiser?
I think we probably
I'm
lost.
not sure which way that would go.
I don't
Yeah,
know.
Do you press the call button if the crocodilian next to you is misbehaving?
You just turn the air on for him and let it roll. Yeah.
There you go. You just blow some cold air on him and
Yeah.
he'll calm down.
He'll calm down.
But there is a growing need for artificial insemination. Now, this is used pretty frequently across lots of species. Bill, you ever eat a turkey?
Yeah, I have on occasion.
Every single turkey you have eaten has been produced by artificial insemination.
Really?
Yes.
Wow.
So those big butter balls with the big breasts and
yeah.
they can't copulate anymore because they have been bred to be the shape they are that we all want
Right.
on our Thanksgiving table. Those animals actually can't physically copulate with their shape, and there is a person who goes along and artificially inseminates every single female turkey.
Wow.
And every turkey you have eaten, unless you go out and shot one yourself, and during hunting season, has been an artificially inseminated turkey. Cows, other livestock, artificial insemination, even across like rhinos and other zoo animals, is actually pretty common.
Wow.
Well, that would make
terms
sense
of-
because it's a lot easier to transport that than it is to transport the animal.
Exactly.
Like you were talking about with the crocodilians. Yeah.
That's what's needed for crocodilians, is the ability to move gametes across oceans and breed these animals so we can maintain genetic diversity. There's been some really unfortunate stories where they have moved adult animals from one zoo to another, and you take these two adult animals that have never seen each other, put them in the same enclosure, and they fight.
Oh yeah.
And there are times where one will kill, the male will kill the female. And so it's doubly insulting because, first of all, you have these animals that are declining in their frequency, and now you do this heroic effort to try to keep their reproduction going, and you lose one. So the ability to do artificial insemination in crocodilians and also across other reptiles like the Yangtze River turtle, that is a critically endangered animal. I think there's only a few left in the world right now, but they were trying desperately with artificial insemination to keep them going, and it just wasn't taking. And there's a lot that I think is not known about the interactions between males and females that lead to fertilization. You could just say, oh, you just sort of, put some of the male's gametes into the female's cloaca and voila. And it doesn't work that way.
Yeah, people don't realize how advanced the rituals. I mean, any
Yes.
species has. With alligators. I mean, they
Yes.
they blow bubbles underneath one another, and they do all kinds of cool things that lead
Right.
up to that. You know what I mean? And it's
They get that whole process going.
They
and,
do.
And if you're just trying to do it artificially, it just may not take because.
So here's something people might not know. So alligators, the male, they have internal fertilization. The male has a phallus. It's pretty rigid, and by muscles, they invert it. It kind of pops out. It doesn't erect. It's not like a mammalian inflatable penis, it's more it's everted out. Muscles contract and it gets squeezed out of the cloaca. But the distal part of it, the end of it, inflates into this really specific structure called a glans, glans. And one thing I've been able to show over the last few years is if you go from crocodiles to Temistoma to caiman to alligators, that distal shape is really species specific and different. They don't look the same. You would assume, oh, crocodile looks like an alligator, looks like a caiman. Well, guess what? At the tip of the male penis, they don't look the same.
Huh.
And that leads
neat.
Yeah, go ahead.
That's neat.
And again, when you see difference in form in nature, that's difference in function. That these things don't happen randomly. So one of the questions myself and colleagues have been trying to figure out is, what do these shapes do? And if you think about the analogy of, say you have, you need to turn a bolt or something like that, you need to go find the tool that fits that bolt you have to move. And you can think about different sizes and different shapes. In a very similar way, the interface between the male and the female crocodilian species has some real specificity. And my hypothesis, one of these, is that really leads into this idea of why artificial insemination in crocodilians doesn't work.
Now,
so
has it never worked, or does it just rarely work?
Very, very. We're talking like single animals.
Oh, okay.
There's been a lot of work done in Australia. There's a Corona ranch, or Corona farm down there. They're working to try to get this going to breed saltwater crocodiles. And they've made some great advancements, but they have not been able to effectively have this process go forward. So there's still things that are being learned.
Interesting.
Yeah.
Wow. Okay.
You can go so deep with that conversation right there alone.
Yeah.
So I'm going to digress from there for a second. You said there was other research you guys were working on right now, and I know you're teaching currently too, right?
Yeah. So I'm at Stevens College in Columbia, Missouri, and I'm also a faculty member at the MU Vet School. And I have collaborations between the undergrads here and the graduate students there. I do work at the Omaha Zoo, and I've got some great colleagues up there that with Jesse Krebs and others there that I work with. What we've been working on recently is the microbiome of the Wyoming toad. And the Wyoming toad is exactly what it sounds like. It comes from lowland areas of Wyoming. But the truth of it is they have been functionally extinct in the wild now for, I should know this right off the bat, let's say many years. Okay. Wyoming toads are only now in populations found in herpetological conservation places in major zoos where they have breeding populations. So Omaha Zoo and other places, they will breed these animals. They have males and females. They'll fertilize. They'll grow up tadpoles.
Now, what caused them to disappear from the wild?
The exact reason, there's chytrid fungus, which is going to be on the tip of the spear, but I almost always feel when you have these extinction events, it's multiple components. It's water quality, it's pollution, it's habitat, and then chytrid fungus, which is part of the worldwide decline in amphibians, was probably the knockout punch for these guys.
There's so many crazy fungus and parasites too that are going around. Some native, some non-native, that are just wiping out amphibians and reptile populations left and right.
Wow.
Some really weird stuff going on.
We've spread pathogens around the world, and all these animals are having to deal with seeing a wider range of pathogens.
Yeah, adapt or disappear, right?
Yeah. Well, the Omaha Zoo and a coalition of AZA and conservation people are really working at trying to bring these Wyoming toads back. But here's the crux of it right now, is they can breed them in captivity. They can produce toadlets. But when reintroducing them, they're not having success. These animals, they're just not creating sustaining wild breeding populations. So they can put them back, but they're not taking.
Is
So
that
we're
due to too many predators that are already there, or I mean, what?
That's an open-ended question. But we don't think it's a predator thing. What we're looking at is what's called the microbiome. So all of us, you, me, everybody, we have microbes all over our skin. And before right now you go and start washing yourself and scrubbing, those are actually really beneficial for you. We are covered with microbes, but guess what? They are symbionts. They are our friends, and we want them on our skin, and we want them in our digestive tract because they take up the spaces where pathogens could colonize. So we're covered with a healthy microbiota. You do not want your skin to be sterile. You will be in very bad shape. You want to have a healthy microbiota. When you hear about probiotics and all of this, it's maintaining that healthy biological flora that we have. The hypothesis that we're kind of running with is maybe because of these frogs being bred in a lab situation, they're not gaining that microbiome that they need to go and be reintroduced into the wild environment.
You're developing sterile toads.
Maybe so. What we've done recently is we have characterized the microbiome of those toads that were breeding at the Omaha Zoo facility. Now, there's another group in Wyoming who is measuring the microbiome of the wild populations that, as they reintroduce toads, they're getting microbiomes. Again, we're not making a breeding population, but you can get an idea of other toads. And the goal of this is to ask, is there a disconnect? Are we returning toads to the wild that have an appropriate microbiome to survive? And the answer could be one of two things. Yes, it is. And now we have to start looking at other things. We have to ask, is it predation? Is it other things? Again, I'm not the expert on this. I collaborate with people who are. But the other side of it can be, okay, if we know we are returning microbiome naive animals to the environment, can we now inoculate them while being at the zoo? Can we give them the microbiome that they need, that when they were released, they are more resistant to environmental predation?
So is there a way to make a makeshift climate, or like maybe even like a sample size area out there in the wild that they can specifically stay in to start testing these theories?
There is some enclosure things that they're talking about. But in terms of the environment, I got a question for you, Bill.
Okay.
You know Florida. You've seen iguanas, right?
Yeah.
What's when a baby iguana hatches? What's the first thing it eats?
The egg. I don't
know.
No,
it doesn't eat the egg.
I don't know, dude. Come on. What do you mean? I'm going to ask you a question about TV. So how many lines of resolution are there on an HD screen?
Everybody imagine. You're a little baby iguana in the egg. You hatch.
Okay.
Yep.
What's the first thing you're going to want to eat?
Something that's close by, right?
Yeah, it could be.
Something easily accessible, I would guess.
Easily, yeah. This ain't going to run away from you.
Is it the yolk?
Not the yolk. You've internalized all the yolk.
Yeah, you've
already done that.
Okay, that's true. Yeah.
So
let's think about this. What do iguanas eat?
Plants.
Do you think as a brand new hatchling iguana, you could just start chomping down on these fibrous?
Probably not.
Probably not.
Sounds like a loaded question to me. I'm going with
no. Exactly. What do you need to digest? Think about like a cow.
You would need liquids.
You would need liquids, but also
you also need some kind of digestive enzymes of some kind.
Yeah. Do cows actually digest the hay they eat?
I don't believe they do.
Well,
do termites digest wood?
No,
he's asking all these questions and making me feel dumb.
Their internal internal stuff. Yes, the biome breaks it all down, right? Yeah.
Okay.
Yeah, they have the bacteria inside that breaks the wood down. So the termite gives the wood to its bacteria in its stomach, it breaks it down, and then the termite can absorb the nutrients. So let's take the one last. So where would you find the microbiome that you would need as a hatchling iguana to get your stomach microbiome going?
They would. That's a great question. I don't know enough about iguanas to have an intelligent answer about this question.
I have.
Be something liquid form, maybe a ditch, or something there that's
no,
local?
Not liquid. It's more solid.
So you're talking about bugs, insects, things like that?
No.
Okay.
You find them. If you look under a tree with lots of iguanas, you're going to find lots of it.
Like poop.
Poop.
Yeah.
Yes.
Poop.
Oh. Okay.
Interesting.
So if you're exactly, so if you are a hatchling iguana, first thing you're going to eat your mom's poop. And you're like, I feel like, oh man. But you think about it.
So if you're in an iguana cage that's been cleaned and stuff, there's no poop there.
Well, you're going to maintain that. Once you seed your gastrointestinal tract, it's there. But when you're born, you've never had this. So you need to seed your gastrointestinal tract with the microbiome that you need to digest the plants you're going to eat.
Wow.
So think about our Wyoming toads. They're being born in this relatively very nice, very well maintained, but pretty sterile condition. It's not totally sterile, but compared to a water body in Wyoming, it's sterile. So this is the idea: is once we identify what the microbiome is or what the environment is, can we inoculate these animals in captivity, let them develop their microbiome, that when we return them, they've got a much better chance at survival.
But now the only way to do that, to figure that out, is if they're like in their natural habitat.
Yeah. Yes.
But they don't exist in their natural habitat.
Well, they're released. They just don't have longevity.
Right. And they don't have longevity because we haven't figured this out.
Interesting question.
I was going to say, how long is the life cycle of one of these toads anyhow?
We're staying on the air until we figure this thing out.
So I have to say we're new on this project. We're going forward, but that's kind of the idea. But again, the idea of the environment interacting with reproduction and variability and success.
Interesting.
Wow.
Yeah.
Yeah, that's a mind scratcher. Yeah, that's really
well,
that is very neat.
Very detective kind of stuff.
It is.
Yeah. You gotta and it's gotta be, and it's gonna be something that maybe you overlooked. And
it could be the simplest thing ever, I guess, in the grand scheme of things.
Maybe so, but the moral of the story is, let your kids roll around in the dirt.
There you go.
Yes.
Everybody needs their microbiome, and what you do, hey, roll around in the dirt. You drop some food, let them pick it up and eat it. They got to build up their exposure to the world. You can't go through your life without avoiding exposure to the world.
Worst thing that ever happened to me, I think it was in high school. They showed us this National Geographic thing with the electron microscopes, and they showed you those mites that live on your eyelashes and all that. And I itched so bad for a week, man. I was like, golly, these things are all over me. And you just gotta be at peace with.
You have an
knowledge.
Understanding with them. Yeah,
right.
Like you all have an agreement.
Yeah.
You are a colony. You are a colony walking around. You're not just you. You're an entire moving assemblage of living things.
And you have to be. I mean, if you want to be healthy, you have to be.
Yeah, if you have a beard, it's 100 times more than that.
There you go. There you go.
You got Rob. You got a possum, the baby possum in there most of the time, don't you?
I do. Yeah. You're telling my secrets, Brandon.
You got a menagerie living in there.
I do. I keep birds in there sometimes, squirrels. Usually possums because now with the extra gray I have in the beard, they blend in just perfectly.
There you go. Nobody even know.
Maybe you could have it, deeded as conservation habitat.
You're right, man. I am missing out on things. I'm so glad we're having this conversation. So there's you have a background also that some people don't know about as an athlete.
Yep.
Tell us about that and what you've brought from that into your work now.
Interesting. Well, if you go back to the beginning of our conversation, that's where we were talking about my little detour. So I spent 3 years as a professional athlete playing football for the New England Patriots. And that was a great time of your life. And it was a dream, but the truth of it is all athletics, especially even professional athletics, doesn't last, a long time. And I found myself in my middle 20s, having to readjust, having to leave something that I've been pursuing for over a decade. And it's just natural. It happens to everybody. There's no way to avoid it. You know what the joke of the NFL stands for.
Yep, not for long.
Not for long, exactly. You got to remember that all the time. So I'm very happy I made my 3 years. And then you eventually, there's someone there to replace you. So, but I really came back to what do I want to do now? And I've just been curious my whole life. I'm a want to know person. And when I was a kid, I was the kid crawling through the sewers. I was the kid coming home with pails full of crayfish that would then die, and I'd toss them in the bushes, and the cats would all come around our house meowing, looking for the dead crayfish. That was what I was all about. And that kind of took a back seat during athletics because there's only so much bandwidth you have in your life. But I decided to go back to that. And I think the things that I brought from that is, first of all, was dedication. A direction in science with training and research. There are ups and there's downs. There's advancements and setbacks, and that's part of athletics. Is you learn from the victories in athletics, but you learn even more from your defeats. You learn what didn't work. You take assessment of yourself, and that's got to be a fundamental component of science. When you are working to discover, you're working to do, you'll have success, and then failure, setback, or something will go wrong. And if you're a person who can't take that setback and learn from it and reapply it, science isn't going to be for you for long. I think there's also a component of teamwork that I brought from it. Especially as an offensive lineman, and the idea that to value and to support your collaborators, your teammates, is something that I bring to today. And again, Rob, we were talking about meeting in Argentina at the crocodilian specialist meeting. Those collaborators there of keeping those bonds and working together for a common goal, I think is something that I have brought forward from that. And
there's a lot of unique communities, you know what I mean? When it comes to research,
very much so.
There's a lot of unique communities. But buddy, I think we got a pretty special one there with our counterparts that work with crocodilians. I was telling Bill this one time. We've had Marissa on the show. We've had John and Jen Bergen on the show. Russ is one of my board members of my group. We're surrounded by so many good people that legitimately would do anything for you at the drop of the hat. I mean, that's just and there's a lot of communities I don't think that are that way in the wildlife world, but the crocodilian community is that way.
And it's not a community about ego.
You're right.
And it's not about ego. It's about commonality and our desires and what we value and the effort we put into it. And it works really well in terms of who I am in my past. And then I think the last one is just tenaciousness of moving towards a question until you finally get a grasp of it. And when you think about an athletic season, you have a preseason, then you have training, then you have the season and the playoffs, and it's just a long grind. And the truth of it is, science is exactly that way. I think the representation of science in the media is always like, Z, whiz, bang, something, something crackles or pops, and then they're like, eureka, and they're like, we're done. And it doesn't work anything like that. Projects takes months to years to finish, to go from the idea of what we're going to do, to planning it, to executing it, to analyzing it, to writing it, to publishing it. That can be a process equal to an entire athletic season plus. And you have to have that tenacity to stay with it, because if you drop that research at any point, it might as well it won't exist, because no one else will know. It only matters when you publish the paper. Just like in athletics, your efforts only matter at the end of the game when there's a final score. If you stop working halfway through the game, there's going to be no victory there. With science, if you stop working halfway through a project, no one's ever going to know what you did. So that idea of taking things to conclusion, being a closer, is something that really I think everybody needs to foster.
Dude, that was epic.
Yeah, that was great. I mean, that's the only way to add to the knowledge is to finish the task.
Yep. Get after it and keep after it.
Yeah.
Till it's done. Well, buddy, I appreciate you so much for coming on and chatting with us today.
This was great.
This has been a great discussion. We'll have to have you back on again to chat about more of this stuff when you're finding out some more answers about the toads and wherever else you're going next. Because I know you're a lot like me. That's just what's next. Okay, we got this. We'll finally figure this out. Now, what are we working on next? What's the next thing we're jumping into? So,
absolutely, always looking at the horizon.
Yep, I appreciate you a bunch, and thank you for all you do. And brother, it's been a blast.
Thank you, Bill. Thank you, Frank.
Oh yeah, been fun.
All right, Bill. Anything else crazy going on this week?
I'm just trying to I got to get to Palm Bay tonight for a football game.
Oh, it's in Palm Bay.
In Palm Bay.
Home game again.
Yeah.
Next week.
Yeah, next week home game.
Okay. All right. Sounds good.
He's gone. We can talk about him now.
No, awesome dude.
Oh yeah, man, that was incredible. Incredible.
He is one of the most intelligent people you could ever be around. It's. I mean, and he doesn't hit you in the face with it. You know what I mean?
Right.
It's just
yeah.
listening to conversation with a guy. You're like, man, this guy's.
And it all just
he's got it
like
going
fell
on.
Out. Yeah, it was
and
good.
It flows. It just flows. Which is the way it should be.
Absolutely.
It's the way it should be. And you can't, another one of those people. And his professor, he was talking about Lou Gillette. Lou was a friend of mine as well, and that was one of those guys that you could not be around the man without him turning whatever situation you were in into a classroom and him teaching you something. So it was just, that's the kind of people that's so much fun to be around, because you're like, okay, your knowledge base is a little bit different than mine, and not only are you just not going to go do something and just ask me to figure it out, you're going to teach me what's going on.
Yeah.
And he passed away a handful of years ago, and the world lost a brilliant man that had done so much. Yeah, he was at University of Florida for a while, and then went up to the Carolinas and was a professor up there, and just somebody we worked with out in the Space Center and a lot of other places. But just
cool.
a brilliant guy. But anyhow, thank you, Brandon, for hanging out with us. Bill, I guess until next week, huh?
Yes, sir.
Well, thank you all for tuning in. I want to give a shout out to Fisher Nissan up here in Titusville. Thank you for all you're doing to help us out with our research and everything we're doing. Don't forget, we have a big event coming up January 19th in Cocoa Village at the Cocoa Riverfront Park in Cocoa Village and the Civic Center. We're going to have talks during the day from 9 till 4, and then we'll have live music outside from 5 to 11. Have vendors, a lot of great organizations and great people involved in that, and it's all free. Just show up and enjoy it. Ain't going to cost you a dime, except for gasoline to get there, and you could always grab a ride from somebody else. Do like Back to the Future, grab a skateboard and jump onto somebody's car. Don't blame me when something bad goes on. All right, so we'll catch you all next week. Thank you for tuning in, and we'll see you after a bit. Bye-bye.